next speaker is Hanadie Yousef. She's the co-founder and CEO of Juvenna Therapeutics, a venture-backed biotech company mapping the therapeutic potential of secreted proteins to develop biologics that prevent, reverse, and cure chronic and age-related diseases. Dr. Yousef is a leading expert in the biology of aging and mechanisms underlying tissue degeneration. Her work has been supported by many awards, fellowships, and grants from NIH, NSF, Spark at Stanford, just to name a few. Under Dr. Yousef's leadership, Juvenna developed a proprietary pro-regenerative protein library supported by AI-enabled drug discovery and development platform. Please join me in welcoming Dr. Yousef.
I think I'm okay. Hi, everyone. It's a pleasure to be here today and be in a room filled with experts in the biology of aging and tissue degeneration. So really happy to see so many faces in person and really looking forward to many more intimate interactions in person in the new year. So I'm here today to share a little bit more with you about what we're doing at Juvenna Therapeutics, and in particular, our really goal to harness the regenerative secretome of human pluripotent stem cells and translate them into regenerative therapies to ultimately rejuvenate tissues in our body. At Juvenna Therapeutics, it's our mission to decode secreted proteins in particular for better human health through a machine learning enhanced protein drug discovery and development platform that is enabling us to unlock the therapeutic potential of secreted proteins, particularly proteins secreted by stem cells, and then accelerate their translation into engineered biologics for a variety of chronic and life-threatening, debilitating age-related diseases. Over the last now about 5 years of R&D at Juvenna Therapeutics, we have not only established a computational platform that enables us to combine in silico, in vitro, and in vivo screening of secreted proteins for therapeutic potential, but we have established our own proprietary library of secreted proteins, as well as perform our own custom protein engineering that has enabled us to date to not only validate the platform by identifying about 33 hits through our library that can promote regeneration of a variety of different tissues in our body, including muscle. We're now looking at lung, liver, and cardiometabolic systems, to name a few. But we also have a lead engineered biologic, a fusion protein that we're developing towards the clinic for a rare muscular dystrophy with an accelerated aging phenotype that I'll touch upon called myotonic dystrophy type 1. And we've been extremely fortunate to secure about $50 million to date in venture capital. We raised a Series A a little earlier this year and actually announced it just a few weeks ago. So, and as well as NIH and CIRM funding to ultimately build upon our mission. And our mission at Juvenna is really to harness the potential of secreted proteins in our body. Secreted proteins are, as we all know, the body's natural signaling molecules. So they're workhorses of our body that induce signaling proteins that circulate in the blood as well as locally in tissues, are responsible for often binding multiple receptors on a variety of different cells, inducing complex signal transduction cascades, leading to desired phenotypic responses. And because they're already naturally evolved to be highly selective for their receptor targets and highly potent, they're often safer and more easily accessible to translate into therapeutics than some of the newer modalities. And so you could think of blockbuster therapeutics derived from signaling proteins such as insulin or human growth hormone. And at Juvenna, we really feel like there's so much more out there if only we really are able to establish a more advanced systematic method of screening these proteins. And so that gets to really the problem and white space we're filling at Juvenna, which is, we know that the human genome encodes for about 20,000 proteins, and of those in particular, there's about 4,000 secreted proteins that could have the potential to be inducing all sorts of disease-modifying or reversing phenotypes. However, there's about 70,000 indications, and so a brute force just trying to match proteins to particular diseases and indications that they could have potential really ends up being, over 1 billion potential links on the map. And so at Juvenna, we have really built a platform to systematically deconvolute this through first focusing in and really kind of breaking this down one by one, where of all the kind of the human proteome, we're focused on secreted proteins. Of that, we leverage, and what I'll be focusing the majority of the talk today on, quantitative proteomics and machine learning techniques to further hone in on secreted proteins that are enriched for therapeutic potential, specifically proteins secreted by stem cells, embryonic stem cells in particular. And so we hone this down about 20-fold to about 1,000 proteins in our proprietary library. And then we really look at what we call organ mapping, where we will identify key drivers of cell or tissue-specific regeneration targeting phenotypes that we believe to be disease reversing. So in the case of really looking and mapping proteins to muscle, there's about 300 muscle diseases, so that's now a 250-fold further reduction in the search space, really leading to about a 5,000-fold reduction. And in building up this platform in this manner, we've already been able to identify over 13 candidates that can drive muscle regeneration to treat a variety of neuromuscular diseases, as an example. And then at Juvena, we got just to kind of start from the beginning, right, from the early days, what got us excited to want to build this map of secreted protein biology? And it was based on some really key findings and observations of my colleagues and I, where we were able to see that proteins purified by human embryonic stem cells that are secreted by them in their secretome and conditioned supernatants not only had the capacity to drive generation of every tissue in the body, an entire human being, but could actually be repurposed to promote regeneration, repair, survival, and viability of a variety of degenerating adult aged tissue types, such as muscle, chondrocytes from osteoarthritic knees, and human cortical neurons. And so when we set up shop at Juvena, the first thing we really set out to do was to actually reproduce the results and the first observations that my colleagues and I made during my early days as a dissertation thesis student in the Convoy Lab at Berkeley. And so we isolated proteins produced by human embryonic stem cells, the conditioned supernatant, and then developed a method to further purify a subfraction of those proteins that were really driving the regenerative phenotypes that we saw. And when we add these proteins onto, for example, muscle precursors, myoblasts, that are isolated either from injured aged mice or even from humans, human muscle biopsies, we noticed that these proteins as a cocktail were really inducing not just the viability, but really immensely kind of proliferation of these precursor cells. And then when we injected these proteins into aged, injured skeletal muscle, we saw this really immense improvement in the regenerative capacity of this injured muscle in aged animals, specifically reducing the fibrosis, promoting more multinucleated fibers and larger cross-sectional area as well. And then when we started looking at what these proteins were actually doing using RNA-seq, and this was done at Juvena by our first lead scientist, Dr. Tak Mai, what we saw was that these proteins as a cocktail were inducing proliferative homeostasis through a variety of signaling pathways that were interacting in an interactome fashion, promoting ECM remodeling, inducing, for example, BMP and TGF-beta signaling, other specific myogenic pathways. And really what this got us, thinking more about was that these proteins of what turned out to be about 1,000 proteins are inducing pathways that are really specific to the age or disease cell that we were treating them for, to induce more homeostatic signaling, target many of the pathways involved in the health of the cell to promote disease-reversing phenotypes that were specific to that cell. In the case of muscle precursors, it was promoting for the precursors homeostasis, improving their metabolic reprogramming, ECM remodeling, but it was different depending on the different cells that we were treating. So when we added, for example, these cocktail proteins onto chondrocyte precursors, we could promote their proliferation and viability. But when we add this cocktail of disease-modifying proteins onto, for example, post-mitotic cells like human cortical neurons in the face of A-beta, we notice that they're not inducing them to enter the cell cycle. They're post-mitotic, but they are promoting their viability, their health, and, for example, in this model, preventing the cell death in the face of amyloid plaques. And so it was these disease-reversing phenotypes that got us excited at Juvena to want to really build a systematic platform and method for identifying key drivers within this cocktail of cell-specific regeneration with trophic immunologic potential that we could then translate into specific biologics for targeted therapies with targeted dosing regimes that were safe and approved then by the FDA. And so the first thing then we set out to do, and this was led by Dr. Jeremy O'Connell, Juvena's CSO and proteomics expert, was first really develop our own method at Juvena to improve the ability to really purify and concentrate proteins secreted by human embryonic stem cells that were enriched for their therapeutic potential. So he did things like, scaling up the production, improving our protein capture techniques, the ability to actually elute these proteins in order to get enough, it was still in the nanogram quantity, but enough protein to then use the best sort of unbiased quantitative mass spec proteomics method out there, which is, or I'd say mass specs, right? TMT mass spec to actually identify the active ingredients. And the way then we established this proprietary library was really comparing two types of different secretomes, and actually many more secretomes, but in a simplistic fashion, we looked at proteins secreted by human pluripotent or embryonic stem cells that we showed as a purified subfraction were inducing these disease-reversing or modifying effects on a variety of different tissues and cells in vitro, derived from primary human cell models and disease models. And then we also compared them then to the same subfraction of proteins secreted by differentiated cells, such as fibroblasts, that in our hands were not inducing these disease-reversing phenotypes, the effect when added onto aged or diseased human in vitro models. And by doing a differential expression labeling through mass spec, we were able to come up with about 1,000 proteins, our first generation RJUV1K library, that are enriched as a cocktail for their ability to drive disease-reversing effects across therapeutic areas. When we then really looked at the proteins and what they're doing in this cocktail, it was a variety of really exciting effects in terms of the mechanism, in terms of, for example, regulating immune system, inducing integrin, TGF-beta, signaling insulin pathways to promote improved metabolism in these cells in particular, just in general as a cocktail. It was actually, as a cocktail, enriched for the ability to promote muscle development and dystrophy, even though this was isolated from the pluripotent stem cell, Secretome, and had factors that were enriched for wound healing, cerebral health, joint health, tendon health, and a lot more. Right, so what we really like about the unbiased quantitative mass spec proteomics is that, unlike things like, a lot of other really cool techniques like microarrays or, somologic aptamer, this allows us to have an absolute or an identification in a very unbiased manner of proteins, enabling us to find things like really interesting non-canonical secreted proteins, proteins with unique post-translational modifications and isoforms, and we even developed a technique to really quantitate the absolute abundance of proteins in this cocktail, really enabling a lot of avenues for drug discovery. And what's exciting too about secreted proteins is there's actually surprisingly a lot that's not known still about secreted proteins as just a class of proteins. In fact, about 42% of secreted proteins have less than 50 citations per protein, and over 30% of predicted secreted proteins have no known binding partners, or we don't know actually what complexes they form. So there's so much out there that us at Juvena are now able to leverage through this compounding database and computationally driven platform for mining the therapeutic potential and mapping secreted proteins to unmet medical needs. So as mentioned, we isolated the library, and then now what we're doing is once we actually identify the proteins in our library, we're able to build this multidimensional in silico map where we're identifying multiple proprietary features and kind of known features such as biochemical features, tissue expression, cell biology, using a lot of bioinformatics and omics data mining to start mapping proteins with known functions and then use this to actually build a model to predict proteins within our library with unknown or previously undiscovered function to then test. As we test these proteins in our assays across different cells and tissues, we're actually continuing to add features and information, really building this database in a compounding fashion, enabling every round of screening to allow our systematic method of uncovering proteins to be a little bit more predictive and quicker to generate hits. And as we grow now the library and platform, we're actually moving away from just human embryonic stem cells and now expanding our proteomic analysis to multiple other types of stem cell and progenitor secretomes in order to really leverage this multidimensional map and information to better inform new therapeutic area selection and predict what these proteins are doing to then test and generate the hits. We then moved on at Juvena, once we established the library, to really build a systematic 5-step process to actually build a pipeline of biologics based on secreted proteins for different diseases. And that included rank ordering proteins from our library using our deep learning models, bioinformatics, and computational techniques, followed by screening them in a variety of different human in vitro disease models to generate hits. Once we generate hits, we actually retrain our models in order to do iterative rounds of predictions, rank ordering, and testing. And then once we generate sufficient hits for a given therapeutic area in vitro, we move on to perform in vivo target validation in natural aged animal models, as well as relevant preclinical disease models, and then move on to lead selection and protein engineering, where we'll turn these proteins either into things like fusion proteins with improved pharmacokinetic properties, and we're also now expanding into antibodies. And so, the way that we also kind of guide our therapeutic area selection is really, it's actually guided by the biology of stem cell signaling. We're looking at diseases and therapeutic and tissues to rejuvenate and what we actually see in almost every tissue in the body with aging is that there is a degenerative loss, right? There is a loss of the cells, the post-mitotic healthy cells, as well as even the ability of our own stem and progenitor cells to regenerate and repair and replace those lost cells. We're also looking at activity in cell therapy trials and addressable by biologic and unmet medical need. And so this leads to the pipeline today, where we have an asset that's the farthest along, JV161. It's a fusion protein that we're developing for an accelerated progeroid-like disease known as myotonic dystrophy type 1. It's the most common muscular dystrophy. We've gotten several million in CIRM funding to advance towards an IND, which we hope to get in 2024. We have a few other leads for the neuromuscular therapeutic area, and we've now generated numerous hits and growing across a few different therapeutic areas with potential for then advancement preclinically as injectable biologics. I'm going to skip, for the sake of time, the information on the lead program, but I would be delighted to share more on the DM1 program and how one of the many proteins in our library that we've engineered now works. But needless to say, we've gotten a really significant, impressive also in vivo data in multiple animal models, where when we map a specific tissue like muscle, for example, that I'm showing here, we don't just test one preclinical model. We look at natural aging, and then ideally, we're trying to find key drivers of organ-specific rejuvenation that can work across numerous indications to then be developed at least for one, but then in the future hopefully be expanded upon. And so for JV161, it works for age-induced tissue degeneration in a glucocorticoid-induced atrophy model, age muscle injury, a Duchenne's muscular dystrophy model, and as well as a myotonic dystrophy model that we developed at Juvena. So in summary, we really went into our Series A this year and expanding the platform, having built the platform around mining human embryonic stem cells, building a secretome, and generating hits for neuromuscular and skeletal diseases, with then engineered leads in the pipeline for a few different indications and one nominated candidate. And we're now really expanding the platform to a few different core therapeutic areas with the goal to really use our platform as a kind of funnel-like process to eventually generate at least 2 more preclinical programs in the next couple of years, as well as numerous hits and avenues for development that we hope to do in partnership with both pharma as well as academic and institutional collaborators. And so with that, I would like to thank our growing and expanding management team and my co-founder, Jeremy, who did a lot of the proteomics and really brought that systems biology proteomics approach to Juvena, as well as really the whole team. And in particular, I showed a lot of the work and early work of Dr. Tak Mai, who joined Juvena after a 6-year postdoc in the Blau Lab at Stanford. We're hiring. We have way more open positions than what's on the website right now. So if you are interested in learning more and potentially joining the team, I'd be happy to talk as well. Thank you so much.
Super interesting data, super interesting ideas there. I do wonder though, so right now you're
Great question, and get this often, right? When you're looking at a cocktail, it is actually multiple proteins really interacting in a fashion to induce signaling crosstalk. And so at Juvena, the way that we're approaching this is, in fact, we have identified multiple combinations of proteins that are acting synergistically or additively for the rejuvenating or therapeutic benefit. But as sort of our first generation biologics, we've identified individual proteins that are or acting sufficiently to really induce a therapeutic readout with a clear then path to translation into market as a single engineered agent. But as we expand the platform, we are now looking at things like fusing active domains of different proteins, and then also leveraging the success of getting issued biologics with the limitations of the clinical and translational path and cost of that, with that success to actually then be able to do more of those combination type therapies as we grow and become more profitable.
Hey, Hanadie, great talk.
Yeah, thank you.
Quick question about the delivery. So in your DN, you're trying to go for the muscular dystrophies. Do you imagine that biologic is going to be delivered systemically or to multiple muscles?
It's systemic. So we're delivering, and have tested this in our animal models, subcutaneously. So we're really envisioning this will be a pre-filled syringe where, much like insulin, you can take it, patients could take it at home, self-administered or by family members. But unlike insulin, which is daily injection, our biologic is already showing the hopeful target product profile of biweekly, so every 2 weeks injection.
So do you see other target tissues affected by that systemic injection?
And we're seeing that, right? We're looking and seeing the bioavailability. We're seeing the target muscles throughout the body getting affected. We're also seeing potential rejuvenating benefits in other organs, and that's actually kind of the hope, right? Is that even though the FDA doesn't really have a process for that a single agent could potentially target pathways across organs to promote better health span. Mm-hmm.